Yes. Good evening, my students. Yes, good evening. Today I'm going to start with a new topic, that is cell cycle and cell division. Cell cycle and cell division. It's a new topic, and as far as the NEET is concerned, how many questions you can expect? 4 questions minimum. 4 questions minimum. Fine. So यहाँ से हम क्या expect कर सकते हैं? 4 questions expect कर सकते हैं. So we can expect 4 questions from this chapter. So this chapter becomes a very, very important chapter as far as NEET is concerned. And what about your normally CBSE में, if you are from CBSE board, then cell cycle, it is for the first time you are studying cell cycle. ICSE से थे, then definitely आप in class 9 I think you have studied about cell cycle, cell division. Fine. Now, so as because for the first time you are getting this information, we are going to go in depth of it. एकदम basic level से start करेंगे. Today we want to give you the basic idea about what is cell cycle and cell division called. But before we start up, I just want to ask you, what is the significance of it? Cell cycle या cell division होने से इसका significance क्या है? What is the significance of that? Significance क्या है? Vishal, any idea? Cell division. A cell divides into 2. Tell me, is it still going on in your body? Yes, Varun. Very good. So cell repair. So cell repair. कहीं पे कट गया. If there has been a cut and 1,000 cells are required over there to heal. So tell me, if the number of cells, so how many cells you require that portion to heal up? You require only 1,000 cells. Now that happens. 1,000 cell ban gaya and it heals up completely. But why you are not having a tumor out there? Requirement was 1,000 cells, but if the number of cells becomes 1,500, so who decides, Swarashish? Who decides how many cell needs to be divided and then the division should stop a bit? Tell me. That means genetically expressed. DNA. DNA decides कितने cell division के बाद cell division बंद करना होगा. That means cell cycle or cell division is genetically controlled. Any issues about this? ये समझ में आया कि नहीं आया? In near future you are going to become doctors. तो आप सर्जन हो, तो आप ऑपरेशन करोगे गॉल ब्लैडर का. कन्वेंशनल मेथड, पेशेंट मांग रहा है मुझे कन्वेंशनल मेथड चाहिए, मतलब आपको पेट काटना पड़ेगा. लैप्रोस्कोपी नहीं करने दे रहे हैं वो. लैप्रोस्कोपी में क्या होगा? कम इन्वेसिव होगा. It's going to have only, you are going to make only 3 incisions. Fine, and you have to cut the particular organ and you have come up with, the patient is going to be ready to be on the normal job next day, from next day itself. But the patient wants conventional method of surgery. So आपको 8 centimeter काटना पड़ेगा gallbladder निकालने के लिए. Now say it requires 2 lakh of cells to form. बहुत कम बोला मैंने. 2 लाख cells चाहिए, जितना आपने काटा वहाँ पे 2 लाख cells चाहिए. तो 2 लाख heal करने के लिए टाइम चाहिए. So you have to give time. And आप वहाँ पे क्या दोगे? You are going to apply T-bact or any type of other ointment. What is so very special about that particular medicine that you are going to apply over there locally? उस medicine में ऐसा क्या है? Think about epithelial tissues. In epithelial tissues, there are 2 types of tissues. One is simple epithelial tissue, and the one is compound epithelial tissue. And the one is compound epithelial tissue. In compound epithelial tissue, there are many layers. बहुत सारे layer रहते हैं. तो उसमें से एकदम top में जो layer है, the top layer is what? Stratum corneum. It's a dead layer. एकदम top पे जो থাকে সেটা dead layer থাকে. তো dead হয়ে নষ্ট হয়ে পড়ে যায়, নিচে থেকে নতুন layer যায়. What is that layer inside? What is the layer inside? Come on. If you are from ICSE, you have studied that. Stratum malpighi. Stratum malpighi. And that is stratum germinativum. So new cell starts originating from that cell, from that layer. So if the outer layer dies off, new layer starts forming in. If this is a compound epithelial tissue, so compound means there are many cells, there are many strata. So this is one of the cell, the upper layer. This is the second layer. This is the second layer. This is the third layer. And just like that, the last layer is this. ये last layer है. So last layer में क्या होगा? Last layer is called stratum, malpighi or germinativum. It forms new cells and pushes the cells over here, and you know the upper layer has become dead. Upper layer क्या बन गया? Dead बन गया. कैसे dead बना? Due to what? As because it is exposed to the external environment, and because the outer layer is exposed to the external environment, it dies off. ये मरे जाए, ये मर जाता है, तो ये मर जाएगा तो नीचे से न्यू लेयर जाकर उसको हील करेगा, and this layer is called stratum corneum. Stratum corneum. Now tell me, how many cells are required over there in the operation area? Say 2 lakh number of cells are required. So 2 lakh होने के बाद 2 lakh यहाँ से जाएगा, regenerate करेगा. So new cells are going to form it by what process? Cell division. Vishal, tell me. In cell division, we are going to study about two. One is mitosis and meiosis. Tell me, if Vishal becomes doctor, what kind of process will take place in the area where you have gone for cholecystectomy? Cholecystectomy is the medical term for gallbladder removal. Cholecystectomy. Tommy means, you know, vasectomy you have studied. Tommy means removal. Cystic means cystic duct. If this is the gallbladder, this duct is called cystic duct. Cystic duct. And this is gallbladder. So removal of the whole gallbladder along with the cystic duct is called chole. Chole means gallbladder. Cystic duct. This process is called cystectomy. So if you go for this cholecystectomy to a patient, what kind of cell division is going to occur? Because cell division, we can classify it into two. One is mitosis and one meiosis. Vishal, tell me, meiosis hobe na mitosis hobe okhane? Meiosis hobe. Got the point? Meiosis to nahi hoga, kya hoga? Mitosis hoga. That means meiosis takes place. Meiosis takes place where? Meiosis takes place in germ cells only. Meiosis takes place in germ cells only. Meiosis takes place in germ cells only. Clear? And what are germ cells? What are germ cells? Testis and ovary are germ cells. Testis and ovary are germ cells. So we have got two types of cells. One is germ cells and the one is somatic cells. So normal body cells में क्या होता है? Mitosis होता है. So what happens in normal body cell? It is a mitosis which takes place in the normal body cells, and mitosis takes place in somatic cells. कहाँ पे बनता है ये? Somatic cells. तो अगर एक सेल है, one original cell, इसमें chromosome number कितना है? How many chromosome number are there? The chromosome number is say 2n. The chromosome number is say 2n. And the amount of DNA is what? Say 2C. The amount of DNA is 2C. So what is C stands for? C stands for amount of DNA. C क्या है? C amount of DNA है. C is the amount of DNA. Now, what is going to happen? The moment this cell divides, it divides into two cells. It divides into two cells. And this is the nucleus. This is the nucleus. Even after division, the chromosome number remains the same, 2n, 2n. Even after division, the chromosome number remains the same. ये था mother, and these are daughter cells. Say for example, this is a mother cell. Why mother? Because meiosis is taking place in this cell. And these are daughter cells. In the daughter cells, what is happening is the number of chromosomes is remaining the same. And what about the amount of DNA? ये 2C था, यहाँ पे भी 2C, 2C. Even after division, the amount of the DNA remains the same. Now, if the chromosome number remains the same, we call this process as equational division. We call this process as equational division. So, what is the other term for mitosis? It is called as equational division. Equational division. So, now my next question is going to be, In animal cell, name the cells where mitosis is very common. Name the cell where mitosis is very much common. Name the cells. That means mitosis is going to occur in those areas where there is maximum wear and tear. যেখানে কাটা ছেঁড়া বেশি হবে, যেখানে সেল ডैमेज বেশি হবে, সেখানেই वहीं पे जहां पे ज्यादा डैमेज होगा सेल में, वहीं पे आपको क्या मिलेगा? माइटोसिस ज्यादा मिलेगा. And tell me those cells. I think you have heard about this cell called stem cell. In animal cell, I think you have heard about this cell called stem cell. We call this stem cell as initial cell also. Who in this class can tell me? Or I would expect some comments if somebody is watching it. Yes. Tell me the initial cell of RBC. Tell me the initial cell of human RBC. Tell me the initial cell of human WBC. Is RBC is a cell or it's a corpuscle? And what's the difference between these two? Is there any difference? Mitochondria is present inside RBC? No. It's not there. That is why they are called corpuscles. You're close, but not completely.
Most of the organelles are absent in RBC.
Most of the organelles are absent in RBC. That is why they are called corpuscles. Any idea? Durba, try. RBC, WBC, and platelet. These three cells are called as formed elements. These three cells are called formed elements. That means these three cells are being formed inside bone marrow. They are formed inside bone marrow. अगर बॉडी में एक भी आरबीसी आपको चाहिए, वो उसको बनना है inside bone marrow. They need to be formed inside the bone marrow. RBC never divides into two. WBC never divides into two. They never show cell cycle, and that's why they are called blood corpuscles, and that's why they are called as formed elements. Whenever your body needs it, it has to be formed inside the bone marrow. And what is the stem cell of RBC? The stem cell of RBC is called hemocytoblast cell. Hemocytoblast cell is the stem cell of RBC. What is the stem cell of WBC? It is myeloblast cell. What is the stem cell of platelet? They are megakaryocyte cells. Megakaryocyte cells. That means stem cell में सबसे ज्यादा क्या होगा बॉडी में? स्टेम सेल जहां पे है, especially they are found in the bone marrow, there is high chances of cell division occurring over there. Number one. Number two, in the gut, right from mouth to anus. So whenever you have food, आपको क्या लगता है? वो फूड जब आपके इसोफेगस से उतरता है और आपके टोटल गट लाइनिंग से जाता है, आपको क्या लगता है वहां पे वेयर एंड टियर नहीं होता है? आप एक मछली बंगाली हो, मछली खाते हो, हाँ, उसका जो मछली का जो वर्टिब्री है आप खा रहे हो, तो कभी कभी फेंकते नहीं हो, पूरा चिबा लेते हो आपको. जब वो जाता है इसोफेगस से, वो स्क्रैच करके नहीं जाता है वो. सो वहां पे क्या होगा? वेयर एंड टियर होगा ना. तो वहां पे क्या होगा? देयर इज अ हाई चांस ऑफ सेल डिवीजन ऑकरिंग ओवर देयर. देयर हाई चांस ऑफ सेल डिवीजन ऑकरिंग ओवर देयर इन द गट लाइनिंग. सो और कहां पे होगा? एक है स्टेम सेल, द सेकंड सेल इन एनिमल. वेयर? इनसाइड द गट. वेयर? इनसाइड द गट. और कहां पे हो सकता है? टेल मी एनी अदर प्लेस वेयर यू कैन एक्सपेक्ट. और कहां पे हो सकता है? मसल्स में. मसल में कब? जब जिम में जाते हो, जब जिम में नहीं जाते हो? एक्सरसाइज करने से. गुड. एक्सरसाइज करने से. जितना ज्यादा आप एक्सरसाइज करोगे, वर्कआउट करोगे, मसल्स डैमेज होंगे. सो मसल्स रिपेयर होंगे नहीं होंगे? तो उसके लिए क्या चाहिए? उसके लिए सेल डिवीजन चाहिए. हाउ कैन यू हैव अ गुड वॉल्यूम ऑफ योर मसल्स, गुड मास ऑफ योर मसल्स? टेल मी दैट. विदाउट डैमेज कैन यू क्रिएट इट? सो आपको डैमेज करना पड़ेगा पहले, उसके बाद रेस्ट लेना पड़ेगा, नहीं लेना पड़ेगा? या 8 घंटा करने के बाद नेक्स्ट वेट करोगे, 2 घंटा रेस्ट लेने के बाद फिर से 8 घंटा जिम में हिट करोगे? Muscle development होगा? No. So you need to give rest so that it repairs. The muscles repairs. The muscle repairs. So repair करने के लिए muscle में क्या होगा? Cell division होना है. Number 1. Number 2. Next cell, tell me the cell where it is very common. The cell division is very, very fast. The place of the body where the maximum mitosis takes place is the skin. आप सब जानते हो snakes they shed off their skin. आप क्या जानते हो आप किस लेवल पे स्किन रिलीज करते हो? स्किन का सेल्स हर दिन आप जब नहाते हो तो आपके बॉडी से वो निकलता है. The dead cells goes out. Yes, you get to see all those cells coming out only in the winters. But throughout the season, the cells comes out from your body. Fine. तो वो cells हर दिन बॉडी से डेड सेल्स निकल जा रहे हैं. न्यू सेल्स वहां से कहां से? स्ट्रैटम जर्मिनेटिवम से न्यू सेल्स बनते हैं. So these are the areas where in animal cells the cell division takes place in a maximal numbers. Who is going to tell me in plant cell, tell me a tissue where in plant cell there is a huge mitosis is expected? Meristem. Very good. Meristems. And what are meristems? We know that plant tissues are 2 types. We have already done in anatomy of flowering plants. Fine. Tell me, meristems are 2 types. Normally plant cells we can classify them into 2. One is meristematic tissue and the one is permanent tissue. Meristematic tissue কয় রকমের হয়? 2 রকমের হয়. There are 2 different types of meristematic tissues. Number 1, primary meristem, another one secondary meristems. And the primary meristems, you know, the primary meristems are of 3 types. The primary meristems are of 3 types. Number 1, apical. Number 2, lateral. Number 2, lateral. And number 3, intercalary. Intercalary meristems. So these are the meristems where here is a huge number of cell division we can expect. So meristems in plant cells, there is a high chance of what? Cell division. Now when we are talking about cell division, so the topic we are dealing with right now is about cell cycle and cell division. Cell cycle and cell division. ये cycle क्यों बोलते हैं सर? Why we are calling it cycle? Why we are calling it is a cycle? Because it's a sequential event. It's a sequential event. Again, I repeat. Why I'm using this kind of terms is there in your NCERT. Sequential events. And mind you, in the options, you are going to get this kind of terms only. It's a sequential event. When we talk about cell cycle में, as because we have already proved that cell division is genetically controlled. Cell division is genetically controlled. So when please, if you are watching it for the learning process, respect. Don't. Who is Mr. Killer? Please kill yourself. Kill yourself better. बोझ कम हो जाएगा. अर्थ में थोड़ा सा मास घटेगा. खुद को मार दो आप. And don't ever dare to send all this kind of message. ये learning platform है. Learn it. खुद सीखो. अपने पेरेंट्स को भी सिखाओ. गलत किए हैं वो आपको जन्म देकर. It's a fault by them. They should know what is their fault. Sequential events. Cell cycle and cell division. Sequential event. So what is sequential event? So cell cycle is a sequential event. And cell division, what is cell division? Cell division, normally we know that it helps in the increase in the number of cells. It helps in the increase in the number of cells. Okay? Now, Cell cycle is a sequential event, and what is a cell cycle? Normally, if you see that, if you know like this, if you just make the cell cycle like this, not the way that the book has given it to you, it is M, it is G, then this is S, and this is G2. Fine. Now over here you can see right from G1 to G2, right from G1 to G2. It takes 95% of the whole timing of the cell cycle. Kitna percent? 95% of the whole timing of the cell cycle. 95% of the document report. Varun, take it out and report. The person who is Vishal, take it out and report. 95% of this whole cell cycle is interface. Is interface. Okay, and 5% of this. 5% of this is the whole cell cycle is mitosis phase, is mitotic phase or M phase. We call it M phase or meiotic phase. This is a M phase. So only 5% is M phase. Only 5% is M phase. Is this clear? So what is being given in your book is G1, S, and G2. These three together is called interphase, and this is what? M phase. So what is interphase? It's a preparatory phase. What is interphase? It's a preparatory phase. It's a preparatory phase where the cell prepares to divide. The cell prepares to divide. Divide करने के लिए all the ingredients required is being given in interface. Is being provided in interface. So it's a preparatory phase. Now, and that is why ये समय ज़्यादा लगता है in Bengali. कुटनो काटते बेশি সময় লাগে না রান্না করতে? কুটনো কাটতে। তার মানে রান্না করার জন্য যা যা ইনগ্রেডিয়েন্ট দরকার সেটাকে ম্যানেজ করতে বেশি সময় লাগে, and that is interface. But for division রান্না করতে সময় খুব কম লাগে. For division it requires a very less of time. Now, what is the time in human culture when human cell is being in a culture medium, is being cultured in a culture medium? When the human cell is being cultured, it was found that it divides into two, and the time that it takes to divide is 24 hours. Is 24 hours. Is 24 hours. Is 24 hours. And within this 24 hours, how many phases are there? There are two phases which takes place. Come on. One is interphase and one is meiotic phase or M phase. Meiotic phase or M phase. Meiotic phase or M phase. Clear? Very good. Now, in NCERT, they have given another example of yeast cell. In culture medium, the time taken for the yeast cell to divide is 90 minutes. Is 90 minutes. And mind you, as because NCERT is everything for us, we have to go by that. So human cell in culture medium, it takes 24 hours. Interface 95% time इसमें ले लेगा. Now what is the time taken? Now you can expect questions from here. The time taken for the G1 is 8 to 12 hours. The time taken for S phase is 6 to 8 hours. The time taken for this G2 is 3 to 4 hours. So if the question is ये सबसे ज्यादा टाइम कहां पे होगा, कहां पे लगेगा? It is G1 first. सबसे ज्यादा टाइम कहां पे लगेगा? For the G1 phase, then S, then G2, and last M. This is very, very important. सबसे ज़्यादा टाइम G1 में लगेगा, then S, then G2, then M. इसमें क्या प्रिपरेशन चलता है? इन तीनों में क्या प्रिपरेशन चलता है? What is the preparation that takes place in over here? We are going to talk about that now. So, yes. Now, the first thing that they have given, is about G1. Let us talk about G1. G1. What is G1 phase? It's called gap 1 phase. It's called gap 1 phase. So gap 1 phase is between what? G1 phase is between M phase and S phase. So for your MCQ, G1 is in between M and S. If this is a cycle, G1 is between M and S. G1 is between M and S. Now in G1, what happens? You're going to understand इसमें होता क्या है. If you understand what happens in S. S for what? S for synthesis phase. S for synthesis phase. किसका synthesis?
Proteins.
Fine. किसका synthesis? Replication. DNA replication होता है यहाँ पे. What happens over here is DNA replication. DNA replication takes place over here. Replication of DNA takes place over here. Now, DNA replication के लिए what are required? What is the enzyme required? There are 7 types of enzymes which are required. The 7 types of proteins which are required. 7 types of proteins, and majority of the enzymes are proteinaceous in nature, except ribozyme, which is non-proteinaceous. And we have done this in biomolecule topic. हमने इसको biomolecule topic कवर कर लिया था कि non-proteinaceous enzymes are ribozymes, but majority of the enzymes are proteinaceous in nature. So replication of DNA के लिए 7 proteins लगते हैं, 7 enzymes लगते हैं. Now the enzymes which are required for replication of DNA, though it is not the right time to talk about all this, but I'm just giving you the name of it. What is the first enzyme which is required for replication of DNA? Is DNA helicase. ये एक enzyme है, तो इसको बनाने के लिए ये लगेगा यहाँ पे. This is required over here. तो उसका synthesis कहाँ पे होने वाला है? In the G1. क्योंकि G1 में क्या होता है? Preparatory phase. Replication. DNA helicase. Then DNA topoisomerase is the name of the enzyme. DNA topoisomerase is the name of the enzyme which is required next. RNA primase is the name of the enzyme or protein which is required. SSB protein, single stranded binding protein, is required. DNA polymerase enzyme, Roman 3 is required. DNA polymerase enzyme, Roman 1 is required. And DNA ligase. These are the 7 different types of enzymes which needs to be formed where? In the preparatory phase. ये अगर S phase में चाहिए तो definitely इसको बनना कहाँ पे है? G1 में बनना है. कहाँ पे बनना है? G1 में बनना है. So G1 में अगर एक सेल को when a cell needs to divide, उसको ग्रो होना है नहीं होना है? उसको ग्रो होना है. तो ग्रो कहाँ पे होगा? वो कहाँ पे होगा? ग्रो in the gap 1 phase. कहाँ पे होगा? In the gap 1 phase में वो ग्रो करेगा. That means a cell when gets inside the cell cycle, तो ये जो सेल है ये हमारे लिए ओरिजिनल है, मदर सेल है, having 2N 2C. ये सेल जब सेल साइकिल में वो जाएगा तो वो कहाँ पे पहले स्टार्ट करेगा? It's going to start from where? It will start from G1. And what is the basic number of chromosomes they have? 2N. अगर ये diploid सेल है. अगर मैं बोल रहा हूं that means कुछ तो यहां पे थोड़ा ट्विस्ट है. What is that twist? When I'm talking about अगर ये diploid सेल है, tell me एनिमल सेल में माइटोसिस होता है डिप्लॉयड सेल में या हैप्लॉयड सेल में? एनिमल सेल में माइटोसिस होता है डिप्लॉयड सेल में या हैप्लॉयड सेल में? Where does it take place? It takes place where? In animal cell, diploid cell. Mitosis takes place in diploid cell, that is 2n number of chromosomes, the cell having 2n number of chromosomes. But normally mitosis haploid cell में नहीं होता है. Normally mitosis haploid cell में नहीं होता. It's not produced in the haploid cell, but with exceptions. Very good. One exception is there, and that is there is a term which we call it as parthenogenesis. क्या कहते हैं उसको? Parthenogenesis. What is the meaning of this? What is the meaning of parthenogenesis? It is formation of life without fertilization. Without fertilization. Formation of life without fertilization. Formation of life without fertilization. Parthenogenesis. Genesis means what? Genesis means formation. In biology, genesis means what? Formation. Partheno means what? Virgin. Partheno means virgin. Can you tell me another term like this? Parthenocarpy. What is parthenocarpy? Formation of fruit without fertilization. Formation of fruit without fertilization is called parthenocarpy. So formation of life without fertilization is called parthenogenesis. And the example is what? Male drone. It's a bee. Male drone. Male drone. Honeybee. It's a honeybee. What happens in honeybee? Let me give you these details. And then you are going to find that in animal cell mitosis normally takes place in diploid cell, but कहाँ पे नहीं होता है? वो haploid cell में नहीं होता है, but एक ही exception है जहाँ पे होता है, वो है honeybee में male drone. That is, and what is the process? The process is called as this. So this is N and this is 2N. ये male है. This is male honeybee having N, and what is N? N is 16. N कितना है? N is 16. So ये अगर 2N है, क्रोमोसोम नंबर कितना है? 32. And this is female. This is female. So ये तो diploid cell है. इसमें क्या होगा? In female honeybee, what is going to take place? Meiosis will take place to form gametes. So it formed 2 eggs having N and N, having 16 chromosome and 16 chromosome. Now, as because this is a haploid cell, male is haploid. और haploid में क्या होगा? Mitosis होगा. That means it is going to take what kind of process will take place? Equational division होने वाला है यहाँ पे. So it's going to form equational division. So it's going to form what? This is the particular gamete produced by the male one. Now consider this gamete is made to fertilize the egg. So अगर ये fertilization हो गया, N plus N क्या बनेगा? It's going to form what? 32, 2N. तो ये क्या बन जाएगा? Female. So fertilization होगा तो बनेगा क्या? What is going to be the sex of this particular bee? Female. But if this egg remains unfertilized, this unfertilized egg is going to become male drone. Male drone. बोलो समझ में आया क्या नहीं आया? यहाँ पे क्या नहीं हुआ? मेल में क्या नहीं होगा? क्या नहीं होगा? क्या नहीं होगा? So this is the only exception. This is the only exception. Clear? But in case of plant cell, it takes place normally where? In haploid or diploid? In both haploid and diploid. How? If you remember, plant matlab, we have talked about the lower plants. In plant kingdom, we have talked about bryophytes, we have talked about algae. Bryophyte, then we have talked about pteridophyte. तो वहां पे algae में अगर आपको याद रहे, say this is the main plant body. The main plant body is this. और ये main plant body क्या है? Gametophyte. याद आ रहा है? The main plant body say is gametophyte. तो ये gametophyte में क्या होगा? Mitosis होगा. It's going to show what? The gametophyte is going to show what? Mitosis. And this gametophyte is going to show mitosis and form what? Form form gamete. ये भी gamete है, और gamete में कितना क्रोमोसोम नंबर है? N. Because the mitosis means what? What is the other term for mitosis? Equational division, where the chromosome number is going to remain the same. अगर N है उसका original, तो वो equal number of chromosome बनाएगा. It is going to form equal number of chromosomes in the form of N only. तो ये बन गया gamete. क्या बन गया ये? ये gamete बन गया. So what it has been formed? It has formed into gamete. So this gamete and gamete from the other is going to join. Gamete from the other plant is going to join and make what? N plus N. N plus N. क्या बनाएगा ये? The first cell, which is called 2N, is what? Zygote. क्या बन गया ये? Zygote बन गया. And in this zygote is going to show mitosis. Is going to show mitosis and form into what? Sporophyte, which is 2N. क्या बनाएगा? Sporophyte, which is 2N. And this sporophyte, ये sporophyte में क्या होगा? ये sporophyte से बनेगा spore, and spore is what? N. ये क्या बना? Spore बना, और ये spore क्या है? N है. तो spore बनने के लिए from 2N to N, what is required? What is required? Meiosis. So Varun, tell me, what is the other term for meiosis? Reductional division. The other term for meiosis is reductional division. Is this clear? Any issues? So American here. Clear? So from this sporophyte, ultimately by the process of meiosis, क्या बन गया? Spore. And this spore is going to germinate to give rise to what? Gametophyte. This spore is going to germinate by what process? By mitosis, ultimately is going to form huge number of multicellular structure called gametophyte. So in plant cell, what you have found? Mitosis and meiosis takes place in both haploid and diploid cells. Mitosis can take place in both haploid and diploid cell. So mitosis can take place in both haploid and diploid cell. Is this clear? Any issues with this? Clear? समझ में आया क्या? Yes? Now, let's get back into that phase called G1. That phase called G1. So, what happened in G1? G1 में replication के लिए, DNA replication के लिए, all the enzymes which are required is being produced where in the G1 phase. And also, the cell shows growth. Cell shows growth. So, in a nutshell, yes, G1 में क्या क्या होगा? The cell grows. The cell grows, number one. Number two, it forms all the proteins or enzymes, required for protein synthesis, required for DNA replication, required for DNA replication. Is this clear? So that's about gap 1 phase. That is about gap 1 phase. Any issues? बोलो कोई questions है? Yes. Any questions? Any questions? Now, so gap 1 में अगर ये क्रोमोसोम है, सोचो ऐसा क्रोमोसोम है. So this is a chromosome. This is a chromosome. This is a centromere. Okay. Now, ये chromosome है. This is chromosome number 1. Say there is another chromosome which is like this. This is the centromere. Another chromosome number 2. And say, for example, there is another chromosome which looks like, say, this. This is the centromere. This is chromosome number 3. So in G1, what you can see, is 2N, 2C. 2N and 2C. But यहाँ पे आपको sister chromatid नहीं मिल रहा है. What you are not getting is sister chromatid. ये बीच में क्या है? What is there in between? You know centromere from the last chapter, cell, the unit of life. Centromere, centromere. This is centromere. And this is what? Chromatid. Now, यहां पे सिस्टर क्रोमैटिड नहीं बना, तो ये G1 में सिस्टर क्रोमैटिड नहीं बना, बट इन G2, what you can see in G2, what you can see is यही जो सेल मैंने बनाया था यहां पे, इसका और एक कॉपी बनेगा. क्या बनेगा? इसका और एक कॉपी बनेगा. And this becomes what? This becomes sister chromatids. क्या बन गया ये? Sister chromatid. ये G1 and G2 में sister chromatid. G1 and G2 में this is the difference. This is the difference. This is chromosome number 1. Say chromosome number 2 क्या था? ऐसा था. Now they are having another one like this. So this is again sister chromatids. ये centromere है. और एक ऐसा बना था, ऐसा बनाया था. And what you can see, there is another one which is called sister chromatid. This is the major difference between G1 and G2. And what happens in G1? In G1, the number of chromosome was 2n, and the amount of DNA was 2c. Amount of DNA was 2c. But in case of G2, it becomes what? 2n and 4c. 2n and 4c. What is the meaning of 4c? That means the amount of DNA has been doubled. यहाँ पे क्या हो गया? Amount of DNA क्या बन गया? Amount of DNA doubled हो गया. The amount of DNA has been doubled. Any issues about this? This is a major difference between G1 and G2. अब बोलो ये 4c कहाँ पे बना? 4c कहाँ पे बना? We know the cell cycle. Cell cycle में अगर ये G1 है तो ये S phase है, तो G1 में, और ये सोचो G2 है. This is M phase. तो यहां पे in S phase, when a cell gets inside the cell cycle with 2n and 2c, जब S phase में आ गया ये क्या बन गया? 2n 4c. क्या बन गया? 2n 4c. The amount of DNA has been doubled over here. That means DNA replication यहां पे हो जाता है. So DNA खुद को क्या बनाता है? खुद का कॉपी बनाता है यहां पे by the help of those 7 enzymes which are produced over here in the G1 phase. Any issues? Come on. बोलो कोई प्रॉब्लम है यहां तक? यहां तक कोई प्रॉब्लम है? Any issues? Yes. Yep. Good. ठीक है, चलो ओके. तो ये रेप्लिकेशन हो गया. अब यहां पे जब G2 में आया तो क्या रह गया इसका? व्हाट इज द अमाउंट ऑफ द डीएनए? इट रिमेन्स टू 4c ओनली. द अमाउंट ऑफ डीएनए रिमेन्स 4c ओनली. पर डिफरेंस क्या हो गया यहां पे क्रोमोसोम नंबर का? क्रोमोसोम का स्ट्रक्चर में दिस इज सेंट्रोमियर 1, पर यहां पे वही क्रोमोसोम क्या हो गया? With two sister chromatid, with two sister chromatid. ये डिफरेंस था ना, ये डिफरेंस था. एंड रिमेंबर दिस डिफरेंस इज वेरी वेरी इंपॉर्टेंट. G1 और G2 में ये डिफरेंस सबसे बड़ा इंपॉर्टेंट है. फाइन. नाउ इन हायर क्लास व्हेन वी आर गोइंग टू स्टडी अबाउट कैंसर, व्हेन वी आर गोइंग टू स्टडी अबाउट कैंसर, क्या जानते हैं कैंसर के बारे में? व्हाट डू वी नो अबाउट कैंसर? अनकंट्रोल्ड सेल डिवीजन ऑकर्स लीड्स इनटू कैंसर. कैंसर कब होता है? Uncontrolled cell division. Come on. Uncontrolled cell division leads into cancer. So definitely, cancer का major reason क्या है? Cancer का major reason है mutation. क्या reason है? Mutation is a major reason. Though cancer is also genetically determined, क्योंकि there are some virus which are oncogenic virus. Oncogenic virus भी है, वो different issue है. जब पढ़ाएंगे कैंसर तब हम देखेंगे. क्लास 12 में we are going to study cancer topic. Now यहां पे G2 में there is a checkpoint. In G2 there is a checkpoint. In G2 there is a checkpoint called G2 checkpoint. तो अगर यहां पे S phase में रेप्लिकेशन हो गया, नॉर्मली वी नो कि डीएनए में अगर यहां पे एडेनिन है, यहां पे क्या बनेगा? थायमिन विद डबल बॉन्ड. यहां पे साइटोसिन है तो क्या किससे बाइंड करेगा? ग्वानिन से विद ट्रिपल बॉन्ड. बट इफ ड्यूरिंग रेप्लिकेशन, जब रेप्लिकेशन हुआ, यहां पे टी के जगह पे ए होना चाहिए, ए ना होकर टी बन गया, ये कॉमन है, ये नॉर्मल है या एबनॉर्मल हो गया? It has been an abnormal case, and this is what is called mutation. And you know what? The major reason for this cancer is TT dimer. The major reason for cancer is TT dimer. तो यहां पे अगर TT होता है रेप्लिकेशन के समय, कॉपी करने के समय अगर TT हो जाता है, तो वहां पे एक एंजाइम है, ड्यूरिंग द डीएनए रेप्लिकेशन देयर इज एन एंजाइम जो यहां पे वो क्या करेगा, टी के जगह पे ए करेगा. व्हाट इज द नेम ऑफ द एंजाइम व्हिच हेल्प्स इन प्रूफ रीडिंग? उसका नाम है डीएनए पॉलीमरेज 1. डीएनए पॉलीमरेज 1 इट हेल्प्स इन प्रूफ रीडिंग. ये किस में हेल्प करेगा? प्रूफ रीडिंग में हेल्प करेगा. इट इज गोइंग टू हेल्प इन प्रूफ रीडिंग एंड चेंज दिस कैपिटल टी विद ए. इट शुड बाइंड विद ए. तो यहां पे टी को रिप्लेस करके क्या बनाएगा चेक करने के समय. दिस इज हाउ इट इज गोइंग टू करेक्ट द म्यूटेशन व्हिच हैज हैपेंड. बट देखो कैंसर कैसे चांस इवेंट बन गया. हाउ कैंसर हैज बिकम अ चांस इवेंट, हाउ कैंसर हैज बिकम अ चांस इवेंट इज ड्यू टू द फैक्ट दैट यहां पे डीएनए पॉलीमरेज यहां पे काम नहीं कर पाया, हैज नॉट बीन एबल टू चेक इट. जो यहां पे डैमेज हुआ है, इस डैमेज को हील नहीं किया. कौन? डीएनए पॉलीमरेज 1 हैज फेल्ड टू करेक्ट इट. एंड सेकेंडली, अगर फेल किया हुआ होता तो सेकेंड टाइम इसको करेक्शन कौन करता? G2 चेक पॉइंट. G2 के बाद वो M फेज में जाने के पहले G2 के यहां पे G2 चेक पॉइंट है. एंड दिस G2 चेक पॉइंट इज डन बाय P53 जीन. व्हाट इज द नेम ऑफ द जीन? इसका रोल कौन निभाता है? अ टाइप ऑफ जीन कॉल्ड P53 जीन. एंड व्हाट इज द फंक्शन ऑफ P53 जीन? शो मी. व्हाट इज द फंक्शन ऑफ P53 जीन? द फंक्शन ऑफ P53 gene is apoptosis. ये P silent है. What is the term? Apoptosis. Apoptosis. And what is the other term for apoptosis? का और एक नाम क्या है? इसको क्या कहते हैं? It helps in programmed cell death. Programmed cell death. मतलब अगर P53 gene G2 checkpoint में रहता है, it remains in the G2 checkpoint. It remains in the G2 checkpoint. And checks whether the DNA which has been produced, जो रेप्लिकेशन होकर आया है, जो कॉपी बन के आया है, वो कॉपी में ठीक ठाक कॉपिंग हुआ है कि नहीं. A is going to bind with C, C is going to bind with G, G is going to bind with C. अगर ठीक ठाक नहीं है तो वो क्या करेगा? It has got a role. P53 का काम है प्रूफ रीड करना फिर से, to check whether the DNA damage is there or not. अगर डैमेज था, टीटी था, तो टीटी को चेंज करके वो टीए करेगा, एटी करेगा. दैट इज द फंक्शन ऑफ P53 नंबर 1. नंबर 2, इफ द डैमेज इज सीवियर, अगर डीएनए डैमेज सीवियर है, इफ द डीएनए डैमेज इज सीवियर, देन इट इज हैविंग द पोटेंशियल, इसके पास वो क्षमता है कि वो इस सेल को यहां पे स्टॉप कर देगा. इट इज गोइंग टू स्टॉप द सेल साइकिल, यहां पे बंद कर देगा सेल साइकिल को. इट इज नेवर गोइंग टू अलाउ द डैमेज डीएनए टू एंटर इनसाइड एम फेज. मायोटिक फेज में उसको आने नहीं देगा. नाउ योर नेक्स्ट क्वेश्चन शुड बी, सर ये कैसे हो सकता है सर? फिर कैंसर क्यों होता है फिर? हाउ कम कैंसर ऑकर्स? हाउ कम कैंसर ऑकर्स देन? मतलब कैंसर इज अ चांस इवेंट. कैंसर इज अ चांस इवेंट. क्लियर? कैंसर इज अ चांस इवेंट. नाउ मतलब दो कोइंसिडेंस एक साथ हुआ. यहां पे टीटी डैमेज हुआ, टीटी बना, डीएनए पॉलीमरेज प्रूफ रीडिंग नहीं कर पाया, इट वाज नॉट बीइंग एबल टू प्रूफ रीड. सेकंड, P53 जीन भी अनचेक छोड़ दिया उसको. देन द डैमेज डीएनए इज गोइंग टू गेट इनसाइड द एम फेज, और एम फेज में जाने के बाद दो डॉटर सेल बनेंगे, इसी का दो डॉटर सेल बनेंगे, और दो डॉटर सेल में अल्टीमेटली क्या बनेगा? द टू डॉटर सेल्स इज गोइंग टू हैव व्हाट? द डैमेज डीएनए इनसाइड इट, एंड दिस इज गोइंग टू प्रोलिफरेट. यही प्रोलिफरेशन बढ़ते रहेगा, एंड दिस इज गोइंग टू लीड इनटू कैंसर. दिस इज गोइंग टू लीड इनटू कैंसर. हैव यू blocked it? Block Rajesh? This guy?
Yeah.
Report? Still he's on. Okay, now what is M? Let's talk about the M phase. Let us talk about the M phase. M phase means meiotic phase. In M phase, how many stages are there? In M phase, you see, अगर ये M phase है, so M phase में चार ऐसे फेज हैं, so कहते हैं प्रोफेज, मेटाफेज, एनाफेज एंड लास्ट टेलोफेज. पर ये चारों मिलके क्या बनता है? ये चारों मिलके क्या बनता है? कैरियोकाइनेसिस. क्या बनता है? कैरियोकाइनेसिस. And the last one is what? Last one is cytokinesis. The last one is cytokinesis. The last one is cytokinesis. So karyokinesis के अंदर कौन कौन आएगा? Karyokinesis के अंदर there are four phases. One is prophase. The second one is metaphase. The third one is anaphase. And the fourth one is telophase. The fourth one is telophase. Clear? That means M phase को हम classify कर सकते हैं into two. We can classify M phase into two. We can classify M phase into two. We can classify M phase into two. क्या क्या? M phase into two. The first one as karyokinesis and the second one as cytokinesis. ये काइनेसिस टर्म कहां से आया? वरुण बोलो. काइनेसिस मतलब डिवीजन और मूवमेंट और डिवीजन, मूवमेंट और डिवीजन. फाइन. किसका मूवमेंट? न्यूक्लियस का डिवीजन और न्यूक्लियस का मूवमेंट. Where from the term kinesis has come? From the term called kinesiology. What is kinesiology? Study of movement and locomotion is called kinesiology. और ये क्या है? cytokinesis मतलब movement of cytoplasm. Very good. So आप नॉट सेल में क्या जानते हो? अगर ये सेल है, this is a 2n सेल, ये मदर सेल है 2n2c. ये जब डिवाइड करेगा तो अल्टीमेटली अगर एनिमल सेल है तो ये क्या होगा? ऐसे बीच से क्या होगा? फरोइंग होगा. What is going to happen over here? What is going to take place? Furrowing. There will be invagination of the cell membrane. और ये बीच का जो न्यूक्लियस है, which is having 2n and 2c, 2n and 2c, अल्टीमेटली क्या होगा? 2n 2c 2c. मतलब डेफिनेटली बीच में कहीं पे ये 2n 4c बनता है, इसलिए 2n 2c दो डॉटर सेल्स में आ जाएंगे. सो ये फरोइंग हो गया बीच से. तो फरोइंग मतलब साइटोप्लाज्म का डिवीजन होगा. तो पहले किसका डिवीजन होगा? पहले न्यूक्लियस का डिवीजन पहले होगा फरोइंग के कारण, या साइटोप्लाज्म का डिवीजन पहले होगा? न्यूक्लियस का डिवीजन पहले होगा. क्लियर? अब सोचो, है ना, ये आपका बर्थडे बैलून है. आप तो अभी बहुत बड़े हो गए हो. ये ऐसा नहीं होता है. अभी जब छोटे थे बैलून के अंदर चॉकलेट था, तो अगर बाहर से ऐसे करके बैलून को आप प्रेस करोगे तो पहले चॉकलेट टूटेगा या पहले बैलून टूट जाएगा? चॉकलेट पहले टूटेगा, उसके बाद जब वो टच कर जाओगे तो बैलून फट जाएगा. सो पहले karyokinesis is going to occur. So first nuclear division is going to occur. Called karyokinesis, and then followed by cytokinesis, followed by cytokinesis. Then any issues? Any issues with this? So 2n 4c, any issues with this? Got the point? Very good. Now, so when we talk about prophase, let's talk about the first phase called prophase. अच्छा, so कैसे याद रख सकते हैं हम? How we can remember all these four stages? By PMAT. P-M-A-T. By PMAT. Fine? By PMAT. Now, what is prophase? Prophase is the longest phase out of these four. It is the longest phase. So it's a question of NEET. NEET में ये question आ गया already. The longest phase in karyokinesis is prophase. And which one is the shortest phase? Which one out of this is the shortest phase? बोलो, shortest phase कौन सा है? Telophase? No, it's anaphase. Anaphase is the shortest phase. Anaphase is the shortest phase. Any issues with this? Now, let us start with prophase. When we talk about prophase, pro मतलब क्या है? Pro मतलब initial. Pro means initial. So initial phase is prophase and the longest phase. What happens over here? अगर ये सेल है, if this is a cell, animal cell, animal cell में ये न्यूक्लियर मेम्ब्रेन है, and we know the nuclear membrane is a double membrane. ये न्यूक्लियर मेम्ब्रेन डबल मेम्ब्रेन है. This is nuclear membrane. This is nuclear membrane. Fine. और ये न्यूक्लियर मेम्ब्रेन है. इसके अंदर क्या है? What is present? Chromatin threads are present. क्या है? Chromatin threads are present. Initial stage में ये chromatin threads यहाँ पे present है. So they have got nuclear membrane, chromatin threads है, and then there are centrioles present. There are centrioles present. Fine. There are two centrioles. There are two centrioles present. ये centrioles है. Fine. Now ये centrioles है. So centrioles present. In the initial stage, these are present inside the cell when the cell enters inside the prophase stage. Fine. So prophase stage में जब cell आएगा, what is going to happen? ये था पहला stage. अब देखो यहां पे, अब यहां पे एक चीज देखो. During the cell cycle, when replication takes place in S phase, सोचो ये अमाउंट ऑफ डीएनए है, ये जब यहां पे आएगा एस फेज में क्या हो जाएगा? द अमाउंट इज गोइंग टू गेट डबल्ड. ये डबल्ड हो गया अमाउंट, बट ये डीएनए किस फॉर्म में है? टैंगल्ड फॉर्मेशन है. दैट मींस दे आर इंटरवाइंड, दे आर टैंगल्ड. दो तरह के क्रोमैटिन होते हैं. वन इज यू क्रोमैटिन एंड वन इज हेटेरोक्रोमैटिन. बोलो, यू क्रोमैटिन और हेटेरोक्रोमैटिन में अगर किसी को डीएनए से प्रोटीन बनाना है, व्हिच वन इज गोइंग टू बी इजियर वन? किसका ट्रांसक्रिप्शन ट्रांसलेशन अच्छी तरह हो सकता है? द वन व्हिच इज मोर इंटरट्वाइंड एंड द वन व्हिच इज लेस इंटरट्वाइंड. दिस इज मोर इंटरट्वाइंड. इसको क्या कहेंगे हम? हेटेरोक्रोमैटिन. क्या कहेंगे? हेटेरोक्रोमैटिन. और ये लेस इंटरट्वाइंड है, क्या कहोगे इसको? यूक्रोमैटिन. टेल मी, इन दोनों में से ट्रांसक्रिप्शनली एक्टिव पोर्शन कौन है? एक्टिव क्रोमैटिन कौन है, यू या हेटेरो? बिकॉज डीएनए से क्या बनता है? अ लिटिल बिट ऑफ इंफॉर्मेशन लेट मी गिव यू. डीएनए से बनता है एमआरएनए, मैसेंजर आरएनए, बाय ट्रांसक्रिप्शन. और एमआरएनए से क्या बनता है? ट्रांसलेशन. इट गोज इनटू राइबोसोम टू मेक द प्रोटीन. सो ट्रांसक्रिप्शनली एक्टिव पार्ट कौन सा है, यूक्रोमैटिन या हेटेरोक्रोमैटिन? यूक्रोमैटिन. तो हमें क्या करना है? ये तो इंटरट्वाइंड है एंड दे आर टैंगल्ड. तो हमें क्या करना पड़ेगा? इसको अनटैंगल करना पड़ेगा. वी हैव टू अनटैंगल इट ताकि वो नेक्स्ट स्टेज में जाकर कुछ करे. तो प्रोफेज में ये क्रोमैटिन अनटैंगल कंडीशन में था. यस. नाउ क्रोमैटिन थ्रेड्स, तो वो क्या होगा? उसको क्या करना पड़ेगा? दैट मींस वो अनटैंगलिंग होने के बाद कंडेंसेशन होगा. सो ये क्या है? ये 4C है. 2N 4C आ गया प्रोफेज में. द सेल विद 2N 4C हैज कम डाउन एंड व्हाट इज गोइंग टू हैपन? Untangling is going to occur over here. Untangling of chromatin threads, chromosomes. Untangling of chromosomes is going to occur. ये NCERT का टर्म है. We have to remember it. Very very important. Fine. Untangling के बाद क्या होगा? Condensation होगा. क्या होगा? They are going to start getting condensed. So क्या होगा? They will start getting condensed. So क्या कहेंगे? Condensation. They are going to start getting condensed and that is what's called as condensation. So यहाँ पे chromatin thread ये अर्ली प्रोफेज में है. सोचो ये अर्ली प्रोफेज में है. जब कंडेंसेशन होगा so they are going to form into this. Nuclear membrane will start disappearing. What is the first step? Nuclear membrane. Nuclear membrane starts disappearing. Starts disappearing. Fine. Acha. What is going to happen to this? What is going to happen to this? Centrosomes. They're going to start moving towards the opposite pole. They will start moving towards the opposite pole. Centrioles starts moving towards opposite poles. Towards opposite poles. Fine. And what is going to happen over here? वो जो क्रोमोसोम था, वो क्रोमोसोम का भी क्या देखोगे? क्रोमैटिन थ्रेड से वो क्या हो गया? अनटाइलिंग करने के बाद क्या बना? Condensation. So when they are going to start condensing, they are going to look like what? They are going to look like chromosomes. They are going to look like chromosomes. This is how they are going to look like. ये centromere है. This is what they are going to look like now. और ये nuclear membrane will start disappearing. Third point, most important, बोलो एंडोप्लाज्मिक रेटिकुलम. अच्छा, और एक चीज यहां पे जानना है, ये जो सेंट्रियोल है, ये सेंट्रियोल के चारों तरफ क्या बनता है? व्हाट इज द प्रोटीन प्रोड्यूस्ड? व्हाट इज दिस स्मॉल प्रोटीन कॉल्ड? These are tubulin protein. These are tubulin protein. अगर ये प्रोटीन है तो ये प्रोटीन बना कब? कहां पे बना प्रोटीन? In the gap 2, G2 में ट्यूबुलिन प्रोटीन बना. अब ये सेंट्रियोल्स और ट्यूबुलिन प्रोटीन, ये दोनों मिलकर क्या बनता है? सेंट्रियोल्स प्लस ट्यूबुलिन प्रोटीन टुगेदर मेक्स इट व्हाट? सेंट्रियोल्स प्लस ट्यूबुलिन प्रोटीन, जिसको माइक्रोट्यूबुलिन कहते हैं. क्या कहते हैं? माइक्रोट्यूब्यूल भी कहते हैं. क्या कहते हैं? माइक्रोट्यूब्यूल भी कहते हैं. दिस सेंट्रियोल्स, दो सेंट्रियोल से क्या बनता है? सेंट्रोसोम. सो इन अदर वर्ड, सेंट्रोसोम प्लस माइक्रोट्यूब्यूल टुगेदर मेक्स व्हाट? स्पिंडल फाइबर. मेक्स व्हाट? स्पिंडल फाइबर. पर स्पिंडल फाइबर के पहले क्या बनाएगा वो? एस्टर. क्या बनाएगा? एस्टर. क्या बनाएगा? एस्टर. सो इज गोइंग टू मेक व्हाट? इज गोइंग टू मेक एस्टर. और एस्टर के बाद ये एस्टर से क्या बनेगा? एस्टर से क्या बनेगा? स्पिंडल फाइबर. क्या बनेगा? स्पिंडल फाइबर. व्हाट इज गोइंग टू फॉर्म? स्पिंडल फाइबर. फ्रॉम एस्टर स्पिंडल फाइबर गेट्स फॉर्म्ड. नाउ दीज टू ऑफ द एस्टर, दो एस्टर प्लस स्पिंडल फाइबर, दो एस्टर प्लस स्पिंडल फाइबर, ये दोनों मिलके क्या बनेगा? ये दोनों मिलके बनेगा माइटोटिक apparatus और स्पिंडल apparatus. माइटोटिक apparatus और स्पिंडल apparatus. स्पिंडल apparatus ये और है. फाइन. माइटोटिक apparatus और स्पिंडल apparatus. ठीक है. सो इन अ नटशेल, न्यूक्लियर मेम्ब्रेन starts disappearing. Centrioles or centrosomes starts moving towards the opposite pole, starts making microtubule. दोनों को मिलकर कहते हैं aster. Two of the aster along with the spindle fiber, they make what? Meiotic apparatus. इसके बाद वो क्या बनाएंगे? वो बनाएंगे spindle fiber. क्या बनाएंगे? वो spindle fiber बनाएंगे and ultimately mitosis में वो help करने के लिए help करेंगे. But along with this, what else is produced? What else starts disappearing? यहां पे inside nucleus यहां पे एक dense substance था. What is the dense substance called? The dense substance present over here inside the nucleoplasm. Tell me, what is the dense substance called? The dense substance is called nucleolus. क्या नाम है? Nucleolus. So what is the third step? The third step is nucleolus also starts disappearing. Nucleolus starts disappearing. Any issues with this? Nucleolus starts disappearing. So 3 terms. Number 1, nuclear membrane starts disappearing. Number 1. Number 2, centriosomes or centriole starts moving towards the opposite poles. Number 3, nucleolus starts disappearing. और हम क्या जानते हैं, न्यूक्लियस का काम क्या है? To produce rRNA, to produce rRNA. अब बोलो, $1 million क्वेश्चन आपको पूछा जाता है, why protein synthesis does not take place in mRNA? Protein synthesis, ये क्वेश्चन, why protein synthesis does not take place in M phase. बोलो क्या कारण हो सकता है? वरुण, tell me. Shronasthi, you can try with this. What is the reason that protein formation never takes place in M phase? In M phase, nucleolus starts disappearing, and nucleolus makes what? rRNA, ribosomal RNA, and ribosomal RNA ultimately is one of them which helps in protein synthesis. That is the reason why protein synthesis never takes place inside M phase. Is this clear? So in the late process, what happens in the late process? The total nuclear membrane disappeared. You can see dense chromosome with 2 sister chromatids, and the centrioles, they have come to the opposite pole. That means the whole these chromosomes are now not present inside the nucleus, but present inside cytoplasm. They're present inside cytoplasm. Any issues with this? Clear? Any issues with this? Got the point? Very good. And what about the other cell organelles? Endoplasmic reticulum. Endoplasmic, 4th term. ER and Golgi bodies. Endoplasmic reticulum and Golgi bodies. It also disappears. Endoplasmic reticulum and Golgi body disappears. There are 4 points that you have to remember about prophase. So 3 phases are there in prophase: early prophase, mid prophase, and late prophase. In late prophase, this is what you can see right now. Clear? So what is our next topic? The next topic is about metaphase. Just wait. This is cell cycle. Yes. So what is the next topic? The next topic is about metaphase. And we don't have time to continue with this. So let me give you this particular idea. Metaphase, we are going to start with the next class. Yes. Let us see some of the NCERT topics which has been given about it. NCERT topic. Yes. All organisms, even the largest start their life form from a single cell. You may wonder how single cell then goes on to form such large organisms, and that is due to what? Growth and reproduction are the characteristics of cells, indeed of all living organisms. Now, even our journey has taken place from what? Zygote. हमारा journey कहाँ से start हुआ? Zygote से start हुआ. The first cell of life is what? Zygote. So from zygote, the day that we have taken birth, after 280 days approximately, what is that is called as gestation period. Gestation period के बाद जब जन्म हुआ, when the birth took place, approximately 10 to the power 14 number of cells were there. 10 to the power 14 number of cells were present over there. So वो किस तरह से बढ़ा? Mitosis or meiosis? What happened? Mitosis. Can you imagine within a span of 9 months, the weight of, when you have taken birth, your mother proudly is to say, 3.5 kg हुआ था मेरा बच्चा. Now 3.5 kg. That is within a span of 9 months, see the number of cells which divided to form the whole body. The mitosis rate was huge then. All cells reproduce by dividing into 2, with each parental cell giving rise to 2 daughter cells each time they divide. अच्छा मुझे बोलो तो ये, whenever we talk about cell cycle, we are talking about eukaryotes. We are talking about eukaryotes. Eukaryote source cell division. Eukaryote source cell division. That is, cell division takes place in both eukaryotes and prokaryotes, bacteria. Cell division takes place in eukaryotes and prokaryotes. Cell division takes place in bacteria and in eukaryotes. These newly formed daughter cells can themselves grow and divide, giving rise to new cell population that is formed by the growth and division of a single parental cell and its progeny. In other words, such cycles of growth and division allow a single cell to form a structure consisting of billions of cells, consisting of millions of cells. This is about the cell cycle. This is about the cell cycle according to your NCERT. According to your NCERT. What it says? What it says? Cell cycle. Cell division is a very important process. Kya ye? Is a very important process. It's a very important process. Cell division is a very important process in all living organisms. During the division of a cell, the DNA replication and cell growth also takes place. देखो, during the division of a cell, क्या क्या होता है? Replication of DNA takes place and cell growth takes place. So what are the two phases which takes place during cell division? Number one, the growth of the cell. Till the time the cell grows to its normal extent, it is not going to divide. That means there has to be some increase in the cytoplasm. Cytoplasm content should increase for it to divide. Okay. All these processes, that is cell division, DNA replication, and cell growth, hence have to take place in a coordinated way. So what is most important term? It's a coordinated way. Sequential event is highly coordinated and genetically controlled. To ensure correct division and formation of progeny cells containing intact genomes. The sequence, I told you earlier also, the sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell, and eventually divides into two daughter cells is termed as cell cycle. So what is a cell cycle? Cell cycle is a sequential event. Cell cycle is a sequential event. Although cell growth in terms of cytoplasmic increase, mind you, the cytoplasm needs to increase. Although in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during one specific stage in cell cycle. DNA synthesis occurs only during one specific stage. What is the stage called, Barun? S phase. What is the stage called? S phase. The replicated chromosomes, that is DNA, are then distributed to a daughter nuclei by a complex series of events during cell division. And these events are themselves under genetic controlled. Most important. So what are the terms we have found over here? देखो क्या क्या terms हैं. एक है sequential event, coordinated way, cytoplasmic increase, genetic control. So write down with a piece of pencil. Pencil दिए लिख रहा. 4 points from this topic. Coordinated way, because these are the terms you will be getting in MCQs. Coordinated way, sequential event, genetic control, and cytoplasmic increase. And cytoplasmic increase. Fine. So next is what? The phases of cell cycle. Phases of cell cycle. In phases of cell cycle, a typical eukaryotic cell cycle illustrated by human cells in culture. These cells divide once in approximately every 24 hours. Human cell, when being cultured in a culture medium, is going to divide within 24 hours. It has been seen. However, this duration of cell cycle can vary from organism to organism and also from cell type to cell type. So, the cell cycle, it varies from organism to organism. Important star mark. From organism to organism and also from cell type to cell type. What is cell type to cell type?
Different type of cell.
What is yeast? What is yeast? If you compare yeast with human cell, tell me the difference. ह्यूमन सेल एंड yeast सेल में डिफरेंस क्या है? सर, yeast is unicellular, sir, and that gives rise to the term called cell type. It's a unicellular one. It is a unicellular fungus, and human cell is multicellular. So cell type to type, there is a difference in what cell cycle. Yeast, for example, can progress through the cell cycle in only about 90 minutes. In bacteria, E. coli cell cycle is of 20 minutes. In bacteria, cell cycle takes place within 20 minutes. That's all for today. Next day, we are going to start with a new topic, and that is right from metaphase. We are going to start. And before that, what NCERT has got to say, थोड़ा clear करके next day, we are going to start with the metaphase one. So very, very important. Go through line by line of the NCERT. Until the time, we have seen the cell cycle. The diagram in the NCERT is very, very important about prophase. You can just have a look. That's all for now. Huh? Can you? Just wait. Just wait. Act second. Act second. Tuesday छुट्टी.